Nā Aotearoa, Naku Tararo Ka Ora ai te iwi. With my basket and with your basket, the people will prosper. And a mana, and a rea aura rakatera mā, tēnā koutou, tēnā koutou, tēnā koutou katoa. Tēnā te raru e te maru o mana whenua, ka te mamoi, waitaha, kaitahu, no mai, hara mai, welcome. I stand here under the umbrella of the people of this place, ka te mamoi, waitaha, kaitahu, and welcome you here to the University of Otago. Ki te whare, tēnā koe, tū tonu, to the house that stands here, welcome, and may you stand forever. But in the case that things might not be so certain about that, if there's an emergency, please use the exits at the side or the rear. If it's an earthquake, drop cover and hold. Ina mate haere hoki atu rā ki te po. To those that have passed, we remember you. Particularly around this time of Matariki. Ko waiau, no Otapati ahau, ko Kotarani te iwi, he kaimahi ahau te whare wānanga o Otago, ko Richard Blakey taka wingawa. Good evening everyone, I'm Richard Blakey, Deputy Vice-Chancellor Research and Enterprise, and it's my great pleasure to welcome you here today to this inaugural professorial lecture, one of the highlights of our academic week as leaders at the university. I bring apologies from our Vice-Chancellor, Professor Helen Nicholson, but welcome the other members of our official party, distinguished guests, and in particular our speaker, David Orlovich. Welcome and congratulations. And I understand there will be a lot of special people in your life here, but also online. So to those attending online and possibly live or in the future, Peter and Phil, your father and mother, Matthew and Susan, your siblings, and maybe even Mark as well, and others attending. So welcome, welcome, and thrice welcome. IPLs are a celebration of excellence at the university from our academic staff, working hard in teaching, research and service to attain the status of professor is an aspirational goal for many, but for those that achieve that goal, we celebrate by inviting everyone together to hear the kinds of work that has led to that. And in David, we've got someone who has been successful in sustained excellence and leadership in all three areas of teaching, research, of service, has been very successful in bringing grant funding to the university, for which we're very grateful, has had extensive national and international networks and leadership, has performed important deep work maintaining collections and databases that are used for many, many people. And it is that depth that is a characteristic of many of our disciplinary experts. But I think what we'll hear from David is how that depth has touched so many lives, quite literally in some cases, for his expert knowledge helping medical professionals, to, in particular in a university context, his depth of knowledge impacting so many students and colleagues. I did a quick tot-up, and there are more than 60 students, 63 students in the CV that were identified being supervised or co-supervised by David. Now, the order of proceedings is a more detailed introduction is going to be given by the Pro-Vice-Chancellor from the Division of Sciences, Professor Richard Barker. We'll hear David, we'll have a summary, and should it all go well, you'll get an invitation to join us at the Staff Club afterwards. So, as I invite Professor Barker up to introduce our speaker, could you all please joining me in giving David our congratulations on behalf of the university for attaining this status of professor. Congratulations, David.
Professor Blakey, tēnā koe. Professor Orlovich, tēnā koe. Dr. Larkham, tēnā koe. Professor Ballantyne, tēnā koe. Friends and colleagues, tēnā koutou, tēnā koutou, tēnā koutou katoa. It is my great pleasure to introduce our speaker tonight, Professor David Orlovich, from the Department of Botany, to give his inaugural professorial lecture here at the University of Otago. As indicated by Professor Blakey, David was promoted to professor in February of this year, and tonight we celebrate this significant achievement. Professor Orlovich was born in Sydney in the small suburb of Monterey on Botany Bay and went to Ramsgate Primary School, then on to Sydney Technical High School. It's always nice to find a personal connection, and the one I've got is that at the time David was a nipper starting primary school in 1971. I was also at primary school up the road that year, a little to the north of Monterey in the suburb of Vaucluse. One of the things I remember about Sydney from those days is just how fascinating it is biologically, and it's certainly a fascinating place botanically. But I wouldn't say that it was a place known for its vegetables. However, David tells us that one of the things that got him interested in plants at a young age was his parents' veggie garden, and he has a distinct early memory of being amazed at how you could plant a bean seed and it would turn into a plant that produced a green bean just like you could find at the supermarket or at the greengrocers. But out of great things sometimes arises disappointment. Young David did not like the taste of beans. Another fascination was a rock garden at primary school, and Sydney is certainly a great place for rock gardens, with the fabulous Sydney sandstone in abundance. And here he discovered that if you paid attention to the growing conditions, you could get plants to grow. This interest in plants led naturally to his BSc Honours from the University of New South Wales before he continued on to a PhD at the same institution, graduating in 1994. This was followed by a succession of postdocs. He went down to Melbourne, then up to Queensland, and then back to New South Wales before we had the good fortune to recruit David to a lectureship here at the University of Otago in 1999. As we have already heard from Professor Blakey, promotion to full professor at the University of Otago is far from easy, and one must excel at each of the academic domains of research, teaching, and professional service. David has been a productive researcher with more than 60 journal articles and book chapters to his name. His track record in attracting research grants has been outstanding, and he's used this to advance our understanding of mycology. Since 2016, he's used a New Zealand $1 million in-kind grant funding jointly with the US Department of Energy's Genome Institute, a community science program to investigate the fungal evolution of fruit bodies and truffle-like fungi. These fungi also led to a Marsden Fund standard grant as a principal investigator. David's interest extends more broadly to plant-fungi relationships, and his 2013 paper in Mycologia excited the global mycology community because he showed that fungal colonisation of the canopy roots of our endemic silver beech provides an unexpected means for increased host nutrition that may have functional significance in forest ecosystems. One of the standout features of Professor Orlovich's research has been the involvement of his students. He has, as we just heard, supervised more than 60 postgraduate students. This includes 20 Masters of Science students and 11 PhD students. And more than half of his published papers have been co-published with his students. This commitment to teaching and students should come as no surprise because it's in his DNA. His mum Phil was a high school English teacher and dad Peter a lecturer in archives administration, and I say hello to both of you. The focus of Professor Orlovich's teaching has been exciting student interest in all things botanical. And a priority has been the incoming students. He coordinated the flagship 100-level paper, Bio 123 and its predecessor, for some 15 years. And just as he brings his students into his research, he brings his research into the classroom. And his students have contributed to a collective biodiversity survey using DNA barcoding and iNaturalist, and in 2022 they discovered a species not known to occur in New Zealand. In 2005 he won an Otago University Students Association Supervisor Award in recognition of outstanding postgraduate supervision. He was also nominated for an OUSA Teaching Award in 2009 and then again in 2010, and our Division of Sciences Outstanding Teacher Award in 2020. In the words of his students, David has a passion for, amongst other things, carnivorous plants and the way understanding these things leads us to fungi. And another student said, I love how excited he gets about his research. It has developed an interest in fungi that I never thought I would have. Evidently, all roads lead to fungi. The third domain in which a professor must excel is service. And David has been head of the department since March 2017. And I believe David was the first head of department that I got to appoint as a PVC. And as part of the process, the members of the department were invited to come and talk to me about who they thought should lead the department. And every single person, except possibly David, said, it has to be David. And can I say that the department's judgment has proven impeccable. David's been an outstanding head of department, not just for the way he's led his own department, but for the way he's contributed to the divisional leadership team. He has many qualities that really come to the fore in those forums. Needless to say, David's passion for fungi is reflected in his service, and a standout has been his organisation of the annual Fungal Foray, a week-long community science workshop which brings together professional and amateur mycologists from all over New Zealand and overseas. David has been a participant in this since 2000, and he has led the Fungal Foray since 2002. He regularly identifies fungi for members of the public, and for those who are risk-averse when it comes to the consumption of fungi, he's also been known to provide urgent consultation for the medical community treating patients hospitalised as a result of their culinary habits. I'll finish there. I think it's now time that we hear from Professor Orlovich himself to hear firsthand about his interests and enthusiasms in all things botanical that have led to this celebration tonight. Please join me in welcoming Professor David Orlovich.
Right. Tēnā koutou, tēnā koutou, tēnā koutou katoa. Ko Hitarari ana, ko Airana, ko Koreiti ana te whakapaparanga mai. Ko poi hakana a Hitararia, te papate whenua, ko David Orlovich aho. Nō reira, tēnā koutou, tēnā koutou, tēnā koutou katoa. Yeah, thank you, Richard, for that. I had to think, are you sure you're talking about the right person? But I guess you must have been. I did recognise bits of it, so thanks very much for that kind introduction. Thanks everybody for coming. Right, I was going to make it all about fungi, but everybody said, oh, you've got to tell them a bit of your origin story as well, so I'll do that. But before I start on that, I have to explain the mushroom that I'm holding, because when you're a mycologist and you've been that for 57, I haven't been that for 57 years, but for a while, people run out of ideas for presents. And so for my birthday this year, I lifted the cover up and there was 3 of these big wooden mushrooms, and I didn't initially exactly know what I was going to do with them, and I had to confess, I said, that's interesting. But then I got an email that said, you got to bring along a prop for the photograph for this, and so instantly I realised they had a purpose. So that's good. And the shirt, I think, was a Christmas present, so that's cool. So yeah, there you go, there's me. That's my primary school uniform, so I guess that was probably 76 or 77, something like that. But I did want to mention carnivorous plants. It has already been said, because I guess I was interested in lots of things when I was a kid, but the plants did really excite me, and that bean story was the taste of the green bean didn't actually turn me off thinking that plants are just incredible. The fact that this little seed can turn into a plant and you can do stuff with it. But the other thing was that I do remember, like, carnivorous plants were pretty exciting in this time, and you couldn't just go to a nursery like you can now, or the warehouse, and buy Venus flytraps, and there are all these beautiful big tissue culture things that are really fancy. But I do remember watching a documentary which was narrated by Vincent Price, who was an old horror movie actor, a really suave-looking dude. And I actually just found it on YouTube, and I spent a good 15 minutes watching it again today. And it's from about 1974, and there was time-lapse photographs of Venus flytraps, and I think that sort of stuff really stuck in my mind, that like plants were cool enough, but plants that actually ate animals and moved, and that was like next level. And then, in high school in year 9, which would have been 1980, one of the science teachers, Mr. Rosca, who was a really funny guy from the country, from Candos in New South Wales, he got up and said at assembly, if anyone wants to start a carnivorous plant club, then come along to the science labs at lunchtime. And so this was like amazing. So turned up, and God, there must have been 50, 60 kids there. And there was no room. And he came out of the science lab into the foyer there and said, I'm not having a club with this many people in it. Go away and come back on Monday with $5. And so we all scurried off and could hardly breathe that weekend. It was pretty exciting, and with anticipation I turned up, and thankfully only about 5 or 6 students came back with the money. And we formed the Sydney Technical High School Carnivorous Plant Club. And, there was a guy in Adelaide who was selling plants by mail order, and so we'd all contribute money and put in an order for these things, and then a week later they'd all arrive wrapped up in Glad Wrap, and we'd share them around. And we'd just meet at lunchtimes once or twice a week in the science labs and sit around going, how's your plants going? And oh, pretty good. How's your plants going? Pretty good. Mr. Rosca was a pretty cool teacher though. He was he wasn't really absent-minded, but he played the sort of the absent-minded teacher, and the students used to kind of make fun of him, but it was his way of getting them engaged, which is pretty cool. And one of the things that also happened was that the Society for Growing Australian Plants, which is now called the Growing Australian Plant Society, I think, they organised a flower show, which was quite a big thing in suburban Sydney at the time. And we got asked if our Carnivorous Plant Club would put on an exhibition. We had a few display tables, and so we all brought them along. And to be quite honest, we really stood out because a lot of the plants we had were not native plants. The Venus flytraps are from the Carolinas in the USA, but no one seemed to care. And we did have some native Australian sundews and the like. And through that, we did that for several years. I got to meet this guy here, Peter Old, as a grevillea expert in Australia. He kind of organised that show, and he won't remember me from a bar of soap, but it was just cool to interact with these people and that were passionate about plants. And as it happened, one year our table was set up next to the National Parks Association table, and I didn't fully understand at the time, but this is it's a group a little bit like Forest and Bird, a sort of advocacy conservation group of people that are supporting national parks in New South Wales. And this guy, Gary Shower, was manning his table, and they were planting little acacia seeds, wattle seeds, and in vermiculite, and giving them away to the kids with a leaflet on how to grow them. And I thought that was pretty cool. And I think when he went off to lunch, I just sort of manned their table for a while, and then he sucked me into joining that society for eventually, which I was in for many years. And again, it was just a little thing, but it just gave me exposure to this sort of environment of people that were interested in the outdoors, and especially in plants. And there was radio, TV celebrities there. I never got to meet them personally, but some of the Australians will know Graham and Sandra Barossa, sort of TV type gardeners. I did actually meet Don Burke at one of them before his TV show became really famous, Burke's Backyard, but he's kind of been cancelled now for possibly good reasons, so that's it. That was a big thing. And then in 1981, when I was in year 10, a really surprisingly big conference came to Sydney, which I didn't know much about at the time, but it was the International Botanical Congress. And this is a really big conference that's got like thousands of delegates, And in 1981 at Sydney University, it was the first time that it had been in the Southern Hemisphere. And it's one of these multi-millions of parallel sessions and things. It's actually the conference where they decide on any changes to the rules of naming plants, but that's probably not why I thought it was that exciting at the time. The previous one had been in Leningrad, and so I was in year 10, deeply into carnivorous plants, and what happened was that some technicians, I think, or two staff, Janice Jacobs and John Pewman at the botany school at Sydney University, organised a youth program for that conference, and it was like a parallel session that school kids came to from all over New South Wales. And it went from the Monday morning to the Friday night, and I didn't know anybody that was any of the other kids that were there, but we all did projects, and I of course did a poster on carnivorous plants, and I took a few of my plants along, and one of them actually got on the TV, which was pretty exciting. And they'd organised for us a lecture series, and they chose the speakers from the speakers at the conference, so there were some really big names there, and so it was a pretty good idea, and I don't think that I've ever been to a conference where I've been aware that sort of thing has been done. It seemed like cool. I do remember this guy down the bottom there is John Pate, who was a Western Australian botanist. He showed some pictures of carnivorous plants, so I knew I was in the right place. And we had a lecture from Peter Raven, who's the guy who wrote the first year textbook that we still use here at Otago, Biology of Plants, so that was cool. And he talked about how you can get aphids to stick their little stylet into the stem of the plant, and they focus in on where the sugar is, where the phloem is, and then they blast the aphid off with a laser, and then they can get the sugar out and measure it. I don't think it was his work, but he wrote the textbook, so he could talk about whatever he wants, right? So, because I think that this was such a big conference to come down to the Southern Hemisphere and be in Australia, it was a bit of a landmark moment for botany in Australia, and so it was quite cool to be part of that as a school kid. The Australian Biological Resources Study had been planning for a few years to launch this series, The Flora of Australia, and they launched the first 2 volumes of that at the Botanical Congress. You can see Australia Post put out a pre-stamped envelope with the first day cover thing there. And funnily enough, one of the students, the school kids that was there, was this guy here who I found on the internet yesterday, he's still alive, called David Banks, and he was maybe a year or 2 above me at school, and he was an orchid expert, and his dad ran an orchid nursery up in the northwest of Sydney, and I think he actually runs it himself now. I knew all about carnivorous plants. He knew a little bit about carnivorous plants, but maybe not quite as much as I did, but he knew a lot about orchids. And we went on one of the field trips to Ku-ring-gai Chase National Park one afternoon, and that's where, at the days, that was well known because that's where they filmed Skippy the bush kangaroo, of course, but it's where they film Home and Away now. Or that area. And David could walk along the track, and I was following him, and he'd go, oh, that's, Grevillea something or other, that's Corea reflexo, that's Epacris impressa, and he knew all the Latin names for all the plants, and that was better than me, and I was pretty damn jealous, I've got to confess. I did get this exposure to people who I thought were kind of more impressive than I was, and I needed to be like that. And David's gone on and written his book on cultivation of orchids, and that's him in Wellington a couple of years ago presenting the Orchid of the Year award to this guy called Merv in Wellington. So from my sample size of 2 people that went to that, we've both kept in our area of botany, which is good. Probably going to jump over a few things here and go straight to my the research that I did for my honours and for my PhD. My main supervisor for my honours and PhD was Professor Anne Ashford, and as it happens, Anne's sister lives here in Dunedin, and her sister's husband is Professor Dorian Owen from economics. So it was a kind of weird connection that I didn't expect I was going to have here. And from very early on in the lectures that Anne gave, she made everything seem really exciting, much more exciting than it probably really was. I don't know, she had this sort of brute force excitement, enthusiasm about everything that she taught, and that was pretty motivating and exciting. My other supervisor for my research work was Guy Cox, and Guy worked at Sydney University and I was at New South Wales University. One of the advantages there was that if a microscope broke down at New South Wales, I could just get in the car, and I had kind of free reign of both of the electrons electron microscope units there. But yeah, the underlying basis for the research that was going on in the lab, or some of it anyway, was that that fungi play a really important role in mediating the communication or the transfer of minerals, nutrients, water even, between the soil and the plants. So there's this saying that's been coined for a long, long time: plants don't have roots, they have mycorrhizas. And the mycorrhiza is this kind of symbiotic organ that is a union between the fungi and the plants. Okay. And so at the time where I was in Sydney, I mean, this is a picture from near Katoomba in the Blue Mountains, but much of the vegetation you can see there is dominated by eucalyptus, and eucalyptus is one of the trees in Australia that forms these mycorrhizal associations, fungus root associations. You can see them in the picture there. The colours that you can see are the fungus growing all around the outside of the root. And in certain times of the year, these fungi that are growing there often produce fruit bodies that come up in the, on the forest floor. And these are mushrooms and toadstools that you might see. So that's a sort of big picture. What do the fungi do for the plants? Well, the main thing that fungi do these fungi do for the plants that was sort of appreciated at the time is that they improve the phosphate nutrition of the plants, especially in a natural situation where native soils are very low in phosphate, and those plants wouldn't do as well without those fungi. They may not be able to grow at all if they don't have those fungi associated with them. So there's lots of interest in understanding how plants survive in the wild by understanding the fungi that are associated with them. New Zealand is the same, and I'll get onto that in a moment with our beech forest. The fungus that I studied for my PhD was this really ugly looking thing called Pisolithus tinctorius. It was known as a horse dropping fungus. So this is the fruit body for it, and I've got to say you would tend to poke it with a stick to make sure it is a fungus before you pick it up. When it's cultured in a petri dish, it's got a nice yellow colour there. I've put that species name in inverted commas there because what it was called when I did the work is probably not what it's called now because there's been changes there. But I'm going to really summarise part of my whole PhD in one slide by just saying that the traditional way of looking at these fungal cells was to fix them with nasty chemicals, aldehyde fixatives, stains, look at them under the microscope so you could see what was going on, put them in the electron microscope, and under the light microscope you see these little red staining granules, and we knew that they contain phosphate, so that's where the phosphate is in these fungal cells. So if phosphate is going to go from the soil to the plant via the fungus, the kind of understanding at the time was that it must somehow be in those granules. Okay, but the trouble is that these fungal cells have cross walls every couple of hundred micrometers, so there's whole palaver with having to try and get the granule. It moves along one end of the cell to the next, and then it must break down and pass through. The phosphate must go through the cross wall into the next one, gets passed along, and it didn't really make that much sense. And so one weekend, really, Anne, my supervisor, was like, well, I was really questioning, are these things actually real? I said, sometimes when I prepare them for the microscope, I don't see them. And she's like, well, prove it. If you don't think they're real, then you've got to show what's really happening. So one weekend, I just sat there looking at the fungus under the light microscope while it was alive, and you could see that those cells didn't really have those granules in them. They're very dynamic. The things move around. The vacuoles are connecting with each other and disconnecting. And what I did was, under the cover slip of the microscope, I flooded all the chemicals that you normally do to prepare them for the microscope. And as I did that, I slowly saw these granules appearing. And so we wrote a paper basically saying that these polyphosphate granules, as one way they were called, are an artefact of specimen preparation, And as it happened, we had a postdoc in the lab called Virginia Shepherd, who was looking at the same organisms, but she was looking at them with a fluorescence microscope, and she'd come up to the lab with saying she'd just been down there looking at these things, and they had all these amazing vacuoles that are moving around, and there's little like strings of pearls and clusters of bunches of grapes was another term she used to describe. And at that point, I'd spent years getting really good at electron microscopy, and it just happened that I had pictures of all of these weird shaped things that she was talking about in the living cells. So again, we wrote some nice papers together describing this vacuole system, and it really was kind of big news when we published that this sort of part of the reason that people had missed all this is because the specimen preparation methods were pretty rough and dodgy, and that really kind of gave this idea that there was a more sensible, workable idea for a pathway for nutrient transport along the fungal cells. Now, I've gone into a bit of that detail there just because one of our students this year, Edie Rogan, is actually going to have a look at another fungus with a similar question, but she's not looking at phosphate transport but nitrogen transport, because there is some evidence that some of these fungi transport nitrogen as well. So that's kind of why I wanted to show you that. Yeah, I guess some of this stuff I just did on the weekend under the microscope was pretty simple stuff, and you do wonder when you do stuff like that, you think, why hasn't anyone else noticed it? And funnily enough, when we wrote the paper showing that these granules were an artefact, I came across a paper in French from 1947 that said that metachromatic granules are an artefact of specimen preparation caused by the precipitation of certain dyes. So we actually only rediscovered something, but it was something that had been not realised. So that's that. The other thing is I had a funny experience. I went to a conference in Belgium in the early 2000s, and I met a Polish professor who said to me that when that paper came out, I was the most hated scientist in Europe. And I thought, oh God, really? I had no idea, to be honest, but she said that it was because they were having trouble getting papers published after that if they'd use these methods for electron microscopy, and that she said to me that one of the resentments was that they didn't have the fancy technology that I had. I can tell you, to freeze those cells, to put them in the electron microscope, I grew them over a bit of membrane filter, and in fact it was the stuff between the membrane filters that stopped the membrane filters sticking together, because someone said try the membrane filters and I didn't realise what they were talking about, so I used the spacer stuff. I grew them over, then I cut them out and stick them on the end of a pencil with rubber on the end of it and banged them onto the copper block that was cooled with liquid nitrogen, and it was all homemade stuff, so it wasn't high technology, but we obviously made it look like we knew what we were doing, so it was kind of cool. And she did say, but after having had the week in Belgium with her, and every beer is a party in a bottle in Belgium, it was like, you're not as bad as I thought you would be. So that was good. Righto. So I just want to very briefly mention that by the time I'd finished my PhD, all the field work I did was going down to the constant temperature room, getting another culture here, subculturing it every 6 weeks. I got pretty sick of that. So, and it just happened to come up that there was a postdoc at the University of Melbourne to look at the development of flowers in the Myrtaceae, the myrtle family, which includes Pahudicava and Rata and Arnica and Carnica here. And I didn't know much about the Myrtaceae. I didn't know much about the evolutionary biology that was behind this. I didn't really know much about floral development, but I certainly learned it. But what I did know was that I was a good microscopist, and they really wanted someone to be able to use a scanning electron microscope. And I should just put a little interlude in there. When I was doing my PhD, at one point my supervisor didn't have any funding to pay for the use of the electron microscope. I'm focusing on the Pro-Vice Chancellors here. And we worked out a deal that I would demonstrate in the EM unit for free, and then in return for that I had free reign of the microscopes. And honestly, for all of the years that I did my PhD, I think my demonstrating was one afternoon a year to the new honours students, and I could be in there at 1 a.m. on a Saturday and Sunday morning, doing the X-ray analyses and things, and I came out of it really well. It was a good deal. It was very generous, but I just can't help thinking that if this was a bit of a user pays thing, I don't think I would have this expertise that I have now. Right? So it's pretty hard and a lot of trial and error, lots of failed stuff. Anyway, so I got that job, and then I learned a lot of scanning electron microscopy. One of the cool things about these flowers is the showy part, as you'll know this from Pahudicava, the showy part in this family are the stamens often, they're not so much the petals. And classification in this family is often on the way the stamens are arranged in the flower. And yeah, really, I guess the upshot of what we kind of discovered is that really it's when you look at these little buds, there's a tiny bud, the thing I've got labelled with a K are the sepals, the green bits around the flower, a couple of petals there, and then the little stamens just starting to form. The way the final flower turns out is really a bit of a function of how much space there is on the floral bud when it's developing, and just really minor changes in timing can lead to very different floral architectures. So that was pretty good fun. I met lots of really great people in Melbourne. I learned a lot about evolutionary biology that I really didn't know so much about, so it was really rewarding. And in fact, these photos are not from Melbourne, but when I came here, I had my first PhD student was Steve Belsham, and he carried on that work with some other groups of myrtaceous plants that included some of the New Zealand natives as well, so that was kind of cool. But really, this is a good day in the office, and so I was introduced to the New Zealand fungal foray when I first got here, and I'll say a few words about that in a minute. But yeah, I guess I have kept up the plant work, and it's good. You can see also with all the background I had why I was sort of I was in a position to be able to teach the basic botany stuff, and I could also teach the mycology, I could do the classification, etc. So that was pretty cool. But yeah, this is David Little in the middle there, who's in the audience here, and Andy Nelson in the red on the right. I think Andy hates the fact that I keep bringing up this photo, sorry. But for some reason I just thought it was a nice photo. That's at the Lake Christabel track, which I think is just south of Springs Junction in the upper part of the North Island. And what we do is really just walk through the forest in the right time of the year, which is often autumn, and look for mushrooms. That's it. And when we find them, we go, yay, aren't they pretty? And if we have permission, which we've been lucky enough to for the much part of our collecting, for all of our collecting, for much part of the time that I've been here, then we will make collections of those and then bring them back here and study them. But I know there's people watching here, and this was put on the Mushrooms of New Zealand Photography Facebook group. There's people here who are far greater mushroom photographers than me, and I just know that, in a way you don't have to be like me and want to know what the name of it is, and to be able to appreciate mushrooms. People just like seeing them and like photographing them. And one of my friends, his wife is in a photographic group, and she said that fungi are always a top topic for people wanting to take photographs of. And so if any of you are interested in this, then there's the Mushrooms of New Zealand Photography Facebook page. Lovely photographs, beautiful composition. They never, nice clean specimens. OMG, idyllic composition. That's pretty cool. Instagram, there are some really good fungus appreciators there. There's an account called Fungifile with an underscore. That's actually Tess McBride, who is a PhD student here in the microbiology department. And her PhD was not on fungi at all. She's doing a postdoc on fungi now, but she's got this beautiful Instagram page with the most incredible photographs. She's postdocing in Sydney now, but so I put those up just because I want to acknowledge the fact that there's lots of people out there who are far better photographers than me, and I just tend to take photos with my iPhone when I'm in the field, and they're not overly, I don't know, when I look at those I think the fungi don't actually stand out that well, they're kind of blending in with the background a little bit. But the advantage of taking a photo on your iPhone is that it's got a GPS tag, so you don't have to forget, you can remember where it's from. And I do try to, get as many views as I can. There's nothing more frustrating than getting a photo where you just see the top of a mushroom and you can't see what it looks like on the underside. So people who photograph fungi know that you really, in order to have a chance at identifying it, even to genus, you really want to see the top and the bottom. So that's why I've turned some of these over, and I've left some standing there. But the field photos I do find they look a bit better, but I'm not as good a photographer as some of the people watching here. So I tend to then, once I've collected these, I'll take them back to wherever we're staying and put them on a grey background with a piece of glass, perspex, and take a photograph with video lights on each side, 4 corners. And Yeah, I find that the lighting in these, it gives me a better ability to, or gives us a better ability, because Andy actually does a lot of the descriptions of our fungi, to be able to see all the features that one might need to see to be able to either identify it, or if we've determined that it's an undescribed species, then to be able to write a description based on the photograph. And in fact, that green one in the centre left there is a species that we described. But yeah, I don't know. There's a few books that have mushroom photos like these, but David Little coined the term dead rat photos. And it's so, which makes me sort of feel like, oh, they're not that good, but because they're laid out like dead rats, you know, but it's kind of stuck actually. Now I just sort of, oh, we've taken the dead rats, yep. So I think they're kind of useful, maybe technical photos is the right way to say it. So then what we do is we will take the dead rat photo, maybe we'll have a field photo as well, make notes about it. This is one from 2014 from a place up near Nelson. We'll make a note of the plants that it was associated with. This is one of these mycorrhizal fungi. Once we've got the collection details, specimen number, photo number, stuff like that, and then we put it on fruit dehydrator, and the fruit dehydrator will basically shrivel it up and look horrible after that, like a dried mushroom that you might find in a bag in the supermarket. The cool thing is that if you dry them reasonably quickly, like on the same day that you collected them, and they're in good condition in the first place, then the DNA remains quite intact in those specimens, and you can go in with some forceps and take off some of the gill tissue and rehydrate them under potassium hydroxide on the microscope slide, and then the microscopy that you need to do on those is totally fine. And in fact, doing the microscopy from the dried specimen is kind of the standard way rather than the fresh, just because then everything looks the same. So that's kind of what we do. Then we type all that stuff into an Excel spreadsheet, save the photos. We will upload them onto iNaturalist. Now, who in here doesn't use iNaturalist? Actually, that's pretty impressive. There's heaps of iNaturalist people. Good on you. Wow. I wanted to intimidate the ones who didn't by making them be the ones that put their hands up. So hopefully while you're watching, I'm going to get boring after a while. So sign up to iNaturalist on your phones. It's sort of like social media for organisms, you know, and you can put fungi on there. You can, if I see a crane fly on the bathroom wall and I'm just getting out of the shower, snap it. Sorry, too much information. And people identify them, you know, moths, all sorts of, it's really amazing. There's your GPS tag, will allow it to be located on a map. Yep, so it's pretty good. Got a scale in there. I will upload a field photo if I have it. One of the things is that if you use a Creative Commons license on there, then people can take those images and reuse them on Wikipedia. And if you don't use that type of a license, like CC BY, then your photo won't be able to be used on Wikipedia. And just this year we had the Botany 365 students, there's a few of them here today, write Wikipedia pages for fungi that didn't have Wikipedia pages, and we were able to troll iNaturalist to get photos off there, which was pretty cool. So there's another 16 fungi on there that weren't there a few months ago. Right, now this does contribute to distributional information, because once two people have agreed that that is the right identification for that species, then they'll get uploaded onto GBIF, which is a global biodiversity information database, and so that becomes a record of where that organism grows. So yeah, I mentioned the New Zealand fungal foray. I certainly wasn't around when it started. But I arrived here in 1999, and the first one I went on was in 2000. We try to go to a different part of the country every year. It was just been in Rotorua this year. It was not on last year because of COVID. Next year the plan is for it to be in the Karamea area in the upper west coast of the South Island. And what we do is we find a base somewhere like a hall, and we set up trestle tables with microscopes. That's from 1986. Honestly, nothing much looks any different. The microscopes are better now. I don't think the dress sense is any different. And we collect in the mornings, and we'll get a permit from DOC. We'll have a list of tracks that we can go to. We'll bring our collections back around lunchtime and then spending time. That's when I do the photographing and making notes about them, talking to other people about what the hell this fungus is that I've never seen before, etc. We generally have a day, the middle of the week, where we have a day of talks, which is a mycology colloquium, which is a really great thing. There's always overseas speakers at that, and really enjoyable things, students telling us about their projects, things like that. When we were up in Northland, that's a more recent one. This was at Opunoni Area School, and they let us use one of their science labs. It was great. And those poor students up there, there was a couple of biology students, but they didn't have a biology teacher, and so they were doing the NCEA online for that, which is pretty hard going. There's the camera set up, straight onto the laptop. There's Park, who's one of the land care scientists who does a bit of the databasing. Some of you might know Jerry Cooper with his back to us there. He's a prominent iNaturalist user and probably New Zealand's best mushroom identifier. And there's our group photo from the Rotorua one, and we were hosted by Scion up in Rotorua, which was a cool place to visit. And yep, we have a fungal foray flag. Right, so maybe now we'll get at a bit of the biological stuff. How are we going? Oh God, not bad. All right, so as you can see, we've been out collecting lots of fungi and trying to understand what sort of diversity we have here in New Zealand. The beech forests, there's lots of fighting at the fungal foray about where we're going to go, and half the people, like me included, want to go to beech forests. Other people are sick of going to beech forests. Why do we have to go to beech forests all the time? And my reasoning is because about a quarter of the species of fungi, of mushroom-forming fungi in New Zealand, are associated with Nothofagus, mycorrhizal with Nothofagus. So it's a pretty high success rate for finding the sorts of fungi that I want to do research on. And the people have estimated, this was an old estimate 20 years ago now, that only about 20% of the fungi that we find in New Zealand overall have a name, have been known to science. And so that is something that kind of motivates me to keep going and to collect more specimens. But if you do the sums then and add up how long I've got left in my working life, it doesn't actually add up. So it may not be possible, but it's sort of one of the things that is kind of exciting to do. So one of the things when I get back from the fungal foray is the people in the department will go, oh, did you discover any new species? And I have to explain that it doesn't quite work like that, because you don't usually pick it up and go, that's a new species, suddenly I'm famous and I've got a Nature paper. Well, you wouldn't get a Nature paper with a new species, no one cares. But the truth is that these discoveries come when you're sitting at home watching Shortland Street and you've got the laptop and some new DNA sequences have come in, and suddenly you can match up the collection you made this year with one you might have made in 2005 in Owakuni and another one you might have made in somewhere else, and suddenly you realise that the DNA has kind of united them all, and that's the exciting, that's when you go, wow, we've got something here, this is cool. So the two parts of how we work out whether we've got a species are we get the DNA sequences and we make sure they're all related to each other on an evolutionary tree. So that's this monophyly. It'd be a bit dodgy if we made one species and the two individuals were totally unrelated to each other. Unfortunately, in the olden days, before they had DNA sequencing, that happened a lot. So there's a bit of sort of tidying up work that we do. And the other thing is that you kind of hope that they'll look the same, and the predictive power of the DNA analysis usually brings together specimens that we forgot actually look the same. There's a blue one with a white cap, and suddenly you sequence one and you look at it and go, oh, it's got a white cap and it's blue, so it all makes sense, so it's cool. So that's how we do it. So one of the exciting discoveries that was kind of took a little while to make this discovery. First, one of our students, Cecilia Wang, sequenced this specimen here, and it turned out to have a different DNA sequence from the common one, which is actually this one at the top right. She gave a talk about that, and some of the scientists in the audience afterwards were like, she's messed that up, it's not right. It's like, oh, I don't know, she's very methodical and she knows what she's doing. So sure enough, she was absolutely right. They're totally unrelated to each other, and in fact over the years since then, this really formed part of Andy Nilsson's PhD. All of these things are quite unrelated to each other, and for 100 years nobody had noticed that. They all gave them the same name at the bottom left there, Quaternarius porphyroideus. And so making a big phylogenetic tree, we were able to separate them out quite nicely. And it's not really obvious because I've only put the purple pouch fungi, as we call them, on the tree. But this one in the middle on the left here, for example, its most close relative is this big brown mushroom. So, and they're all like that, right? So it's not like we've split up one purple thing into, just to make it annoying for everybody, into 6 different things, right? These are 6 totally unrelated things that need to have new names as a result. So we've done a bit of that. Some of them have turned out to be very rare. That top left one's only known from the East Harbour Regional Park near Wellington. Others, like this one on the middle right, from North and South Island, quite common, etc. When we described the middle one on the left there, that one, it was only known from 2 collections up in the Golden Bay area. And so, you know, that's something that you could sort of interpret as being fairly rare, or at the very least you could say is data deficient. We don't really know how, you know, what to make of the commonness of this species. So we've got a solution for that. I'll just mention that one of the other things that I'm trying to do is to sort of help people out by turning our publications of these new species into Wikipedia pages, so you can kind of, you know, the public's got a more accessible way of sort of being able to key them out, and it's quite easy to do that and quite good fun. And whenever I've got an important deadline to do something that I don't want to do, I don't do it, I just do another Wikipedia page. But one of the problems is that our trips to collect fungi are so few and far between. Once a year is not enough. And, you know, they come up when they want to come up, and if the weather's wrong when you got there, then you won't see it, right? So what we've done is adopted a technique that was developed in Germany in the 1990s, where they buried little nylon mesh bags in the soil and filled with acid wash sand, and you leave them there for a few months, and sure enough, these mycorrhizal fungi, the fungi we're interested in, grow into the bags, and the DNA from those bags can often contain up to about 60% of the DNA belongs to these fungi we're interested in, right? So it's actually enriching the sample. If you did that for the raw soil, it would be much lower fraction of those interesting fungi. And so then you get the DNA sequences. Thanks to the next generation sequencing, you can sequence everything in the bag. We can then compare it with other sequences on GenBank, the big database. And in this case, this is a real sequence that we've actually managed to match it back to 100% identical to one of my dead rat photos. So it's cool, from the herbarium here. So you can pick, and you know, if you can detect COVID in wastewater, then you damn well can detect fungi in soil. It's the same sort of idea. So then when Laura Van Galen came to us about 2017, 18 maybe, one of the things that she came from Tasmania. She's now doing a postdoc in Switzerland and is watching. Hi, Laura. When she came to us, we said, well, wouldn't it be cool if we could bury the hyphal ingrowth bags all up and down the country in beech forest and survey what's really the widest survey that's ever been done of the environmental DNA of these fungi that we're interested in. And she's such a capable student, she's just like, yep, that's good. So she buried over 4,000 of them. She coordinated all the field teams of helpers to do that. And every one of those yellow dots represents 10 plots, and at each one of those plots there were 5 hyphal ingrowth bags. So some of them got dug up by people who thought they were, you know, the government or whatever, but that's fine. And so then we sequenced everything in there. And I should say, Laura's done a lot of ecological analysis of this, and that's really her story that I'm not going to tell you, but she did find some interesting associations between fungi and their hosts, and also fungi and temperature, and fungi and nutrients in the soil too, if I remember correctly. But this fungus that we only knew from Golden Bay, we thought, well, where's that? You know, and sure enough, it's come up. These are yellow dots are where the samples were. The red ones are where we detected that fungus, and we're basing that detection on the relationships in the DNA tree. So, you know, we can pretty confidently say now that, in fact, where we thought it was just based up in Golden Bay, it's actually damn well growing in Waipori Falls as well. So it's cool. As well as that, we actually found little clades or groups of sequences for which there was no specimen, no dead rat photo, it doesn't match anything. So there's a species that we know exists, but we haven't found it yet. So this is giving us a framework for being able to discover things. I got the Gene 312 students last year to sequence some of our samples from Piano Flat, which is this place here, just about an hour north of Gore, not far from Roxburgh as the crow flies. We found these beautiful little blue coloured mushrooms with a brown top. We sequenced them. We had a look at where they actually came out in our evolutionary tree, and we found in the environmental DNA they're dispersed all up and down the country, the South Island. And we also found that it occurs in Tasmania. We found a link to a specimen in the herbarium. Well, it's actually in the Melbourne herbarium, but it's from Tasmania, from a PhD student there. So they are currently on the way here in the post. We are pretty sure that this is an undescribed species, and we're going to describe it, and it'll be a species, I'm expecting, given the monophyly here, that it'll be a species that grows in New Zealand and Tasmania. So that's kind of how it works. That one that only grows up in the Wellington area, East Harbour Regional Park, its closest relative is a really rare fungus. It smells like rotting fish, only collected once in 1967. Nobody actually knows exactly where this Kowai bush, and it's not the one near Springfield for the 100th time, is. It's in the Mount Grey area. We believe it may well be farmland now, so it's the closest relative to this rare thing in the North Island. So again, where did it occur? We found it in one of Laura's samples, and it's a couple of 100 kilometres away from where we think this other site was, and near the Mount Somers car park. So there's a destination for a field trip to rediscover this rare thing that we didn't know really where it came from. So that's pretty exciting. We're scraping the bottom of the barrel there, because on our DNA barcoding, it's only one base different, but I still think one nucleotide difference is worth a field trip, 100%. And we can, this Amanita muscaria, the red fungus with the white dots, that's a bit of an invader here. It was introduced into New Zealand on pine, it's spread through the South Island on Pinus radiata roots. It's quite widespread in beech forests, and it's been known to invade and can potentially exclude our native fungi, so we can see where it is. It's all over the place. Right, and so Sam Lasham, who's just finishing his masters, he took all of this environmental DNA data for this big genus called Cortinarius, and he tried to work out, well, how many species actually are there genetically, even if we haven't discovered them all yet. And believe it or not, about 80% were undescribed, which is exactly what those people had predicted 20 years ago. So amazing. All right, so a lot of work to do, but now we have a genetic framework, which is cool. Just want to mention that Daniel Munro, who's a great artist from here in Dunedin, I think based at Port Chalmers, used these fungi as a piece for some stained glass, which is in the Division of Sciences. So I don't think it's actually hanging up anywhere yet, but you can bug Richard Barker about that if you'd like to see it, and if enough people do, it's stunning. So if you do that, then that will be kind of exciting. So just to sort of finish up, I will just finish up by, some people are wondering, well, why are there so many of these things that actually look the same, that aren't related to each other? And in fact, there's a whole lot of these fungi that look a bit like mushrooms, but the caps don't open, and we call them truffle-like fungi. Sometimes they have big stalks, other times they don't really have a stalk at all. Why are there so many colourful pouch fungi in New Zealand? This is a kind of a topic with lots of discussion going on about it. In North America and in Australia, these truffle-like fungi, as they're called, are the sole diet for some of the little burrowing marsupials, things like that. And, like real truffles, which are animal dispersed as well, people have sort of considered this idea that mammals in some of these countries may be dispersing those fungi. In New Zealand, we don't have the grazing mammals except some of the bats. And Dr. Ross Beaver in the mid-1990s proposed that because we have so many ground-dwelling birds that sort of replace the role of the mammals here, then maybe they are responsible for driving the evolution of these. But he did also say in there that it could be other animals as well. One of the things that people have suggested in the literature is that if you're a truffle-like fungus, then one day you wake up and you're in the poo, literally. And so when the spores germinate, then who are they going to find? They're going to find themselves, another spore germinating from the same fruit body because the animal ate it. On the other hand, if you're a mushroom, then you're like an umbrella and you've evolved to be able to disperse the spores via the wind, so they've got a better chance of outcrossing. And so people have suggested in the literature that maybe the truffle-like fungi are inbred. But, I gave a talk about 20 years ago, and Bill Lee from Landcare asked me, why are the mushrooms so colourful? And I go, I wouldn't have a clue. Don't know about that, But then why are the truffles colourful? Oh, it's because of bird dispersal, they like the colours. Why are the mushrooms so colourful? Don't know, I don't know. But we had this dilemma that's a chicken or the egg thing, because if the animals dispersing the truffles drove them, then how did the first truffle form, because it didn't have a disperser yet. And then it kind of got me wondering if maybe the animals are eating the mushrooms too, which they do. They're a good food source in the forest. Then maybe some of those that tend to be favoured by animals maybe then kind of tend to just keep getting eaten, and so they'll tend to become more inbred. The other thing is that in some mushrooms there's been a study that showed that in mushrooms that are self-compatible, they can mate with themselves. If they continually inbreed them, then you know what they start to do. They start to mutate and not really form proper mushrooms, and guess what they look like? The truffle-like fungi, right? So I think that kind of gets us out of the chicken or the egg loop by thinking, well, maybe it started by consumption of mushrooms by some sort of animal leading to the evolution of the new species. So our Marsden isn't really trying to untangle the question of how it all happened in the first place, but looking at the genetic consequences of this. And I don't want to bore you with any of that, because it's been really hard actually. Okay, so I just want to leave you with this message that these fungi are crucial for our forests, our beech forests, because if you plant a beech seedling in a pot, it won't grow unless the fungi are present that can form these associations in the root. But beech trees need to move around the country. In the last ice age, people think that all of Fiordland was covered with ice. So there's no forest there, right? Only 15,000 years ago. So people have wondered, well, where the hell did all the beech trees come that cover Fiordland now? Did they come from the further north? Did they hang around Dunedin or the Catlins and then move across after the climate started to warm up? I was lucky enough to be involved in a project with Nick Rawlins and several students, including Ben Potter there, John Waters, Michelle Knapp from Anatomy, and we did some genetic testing on the silver beech trees, and we found that those ones in the southern South Island are genetically isolated from the ones in the northern South Island, and so that does give a suggestion that it's most likely that Fiordland was recolonised by the trees that were hanging around near the coast, right, in the last ice age. But the trees can't move without the fungi, right? And I've just told you about the fungi needing to move. Some of them potentially need animals to move if they're being dispersed by the animals that are eating them. And it's actually already been suggested in the literature by others that there's likely to be a three-way relationship between the trees need the fungi need the animals to disperse them, and the animals are eating the fungi, helping the fungi to spread, which then provides the inoculum for the trees to spread. There's actually a parallel with wilding pines. Some scientists at Canterbury University showed that introduced possums and deer spread the fungi that help the wilding pines to spread. So it could be good on the native side and bad on the introduced side. So just one of our students, Josie McGovern, who's just finishing up her Masters now, she went up to Orokonui, and they've been very grateful, we've been really grateful that they've allowed us to sample the poo of the takahe that are up there. And what Josie did when she did her postgrad diploma of science is she tried to culture the fungi out of there, and believe it or not, she cultured mushrooms, these things, so those takahe are around there potentially eating, whether it's deliberately or not, the spores on the grass or deliberately picking off the mushrooms, and they're coming out in the poo and they're viable. And so that's evidence that these birds can disperse some fungi. That's actually a wood rotting fungus. But when we did the DNA analysis of some of these dung samples, we also found DNA for mycorrhizal fungi, the fungi that are growing on the eucalyptus trees up there at Orokonui. So she's gone back now and done a much more thorough study, which she's just sequencing all the results for now. So I just want to leave you with this idea that we're discovering the species of fungi that are out there. We've got the ability to map them now. We're interested in understanding what the relationship is between the host of these fungi and the fungi themselves. Some fungi may be able to blow from the South Island up to the North Island and back again, but others may not be able to, so we haven't really looked for patterns there. And we're interested in potential dispersers of these things as well. So the take-home messages are, for the couple of people that aren't on iNaturalist, to sign up for it. Use a good license so you're sharing your photographs. If you like fungi, then embrace it and geek out and join Facebook. Our trees, our fungi and our animals are interconnected through space and time. Okay, they're all part of the important part of the picture there. And the fact is, a lot of the animals that may have evolved with our trees and our fungi are extinct now, so that's kind of sad, but I think that maybe is part of the story. There's a lot to discover in terms of fungi, and that means that when you email me tomorrow with a photo of a mushroom you found in the backyard and I don't know what it is, then it's not my fault, okay? Remember, 80% unknown, okay? And if you really enjoyed this, and for those of you that can, come and study botany, and for those of you that just would like a week away, then come and join the New Zealand Fungal Foray next year. So I'll just finish by thanking everybody, thanking my family, thanking my colleagues in botany and elsewhere in the university and everywhere. And most importantly, thanking all the students, because I'm coming in with the enthusiasm and they're all doing the hard work. So yeah, it's been quite a privilege, and thank you to the university for the honour and the privilege to be able to rave on for an hour like I have. So thanks very much.
Tēnā koutou katoa. Firstly...
Who are you?
Or Matt Larkamaho. I was going to say firstly, tēnā koe, Orangi Orlovich. So can we give him another round of applause, please? David, it was a real honour to be asked to come and say a few words on the behalf of the Botany Department and respond to your talk. And I think everyone here will agree that it's really easy to see why David's been so successful in promoting the importance of understanding fungal diversity. His enthusiasm and obvious passion for his science is really inspiring, and it's contagious. And I myself haven't actually been immune to that. When I arrived here in 2016 as a postdoc studying forest trees, I had no real interest at all in fungi. But now, after 7 years working and collaborating with David, I can't walk into a forest without thinking about what species of mycorrhizal fungi are tapping into the roots below the soil. And actually, my best research outputs are now on the fungal diversity of our forests. So thank you for opening my eyes. But I think, for me, one of the most important aspects of David's research is that it really highlights the critical importance of taxonomy as a discipline. And taxonomy tends to get a bit of a bad rap. It's not the sexiest of the sciences, but it is critically important because it provides the basis for the rest of biology depends. Okay. And David, I think you've shown multiple times now, and you did certainly show tonight, that by continuing to catalogue that diversity and even in these cryptic organisms, it really gives us new insights into the ecology and evolution and the conservation of our biology. So I think we owe you a gratitude for maintaining the cause for taxonomy. But you're more than just a researcher, like all good professors probably are, I think. You're an inspirational teacher, a passionate science communicator, and a leader. It's not an exaggeration to say that students universally love David, and I think that's demonstrated here with so many of them in the audience tonight. David's been, as Richard said, David's been nominated for multiple undergraduate teaching awards. He's supervised over 60 postgraduate students and been recognised for excellence in his postgraduate supervision. David also has probably the hardest thing that he's done in his career, I suspect, is be the head of the botany department for the last 6 years. But he's managed to develop an inclusive and a vibrant departmental culture. For example, I don't think there are many Christmas parties where you go out to the pub and the HOD and other senior academics like Paul Guy are both in the band, and they're actually pretty good, and pretty much the whole department's on the dance floor by the end of the night. So
I'm glad that's the only story you told.
But David's also he's also navigated the department through some of the most challenging periods of our time, and I think it's his personal attributes that have made him such a successful leader through those difficult times. Despite his obvious brilliance, David is always humble, he's always personable, he's unassuming, he puts people first, and he leads with compassion. And in fact, I think that this university would be a better place if there were more people like you around, David. So, without further ado, I'd like to thank you on behalf of the Department of Botany and myself. Congratulations, Professor Orlovich.
Thank you.
You deserve all of your success.
Thanks very much.
Here's a small gift.
Thank you.
Yep.
Great.
And I just needed to extend a welcome to everyone over to the staff club for drinks, and thank you all for coming along. It's a great turnout. Yep.
Thanks very much.